So I think we're going to go ahead and get started. If you'll take your seats, please.
Yeah.
So we're going to begin our discussion session for session number 2. As we did earlier with session number 1, this is an opportunity to talk through some of the specific questions and issues that get at the application of the type of data that we've been talking about, recognizing that we're still early. This is emerging information. What are the kinds of things that might allow us to do the right kind of research or answer the kind of questions that risk assessors and regulators are looking for? This issue of how we deal with inter-individual variability has been a very difficult issue for all three of those communities, and overlaying the idea of the microbiome on this issue of inter-individual variability and how that might affect how we look at these types of data is the subject of this discussion. So we're going to start with a discussion that has to do with the kinds of things that we've heard about in these talks, some of the preliminary evidence that we have, and what that might suggest about whether microbiome research can help us to identify susceptible individuals or populations that would be helpful in terms of the process of understanding impacts of environmental exposures. We have a good group, including our two speakers here. You've met Yvonne, so I won't ask him to introduce himself, but John, would you just introduce yourself?
Thank you very much, Bill. I'm John Vandenberg. I'm with the EPA's Office of Research and Development. I'm the director of our Human Health Risk Assessment Program. And I might note two things. One is that my training was in biophysical ecology, which some of the presentations earlier today and through the afternoon have really resonated with me in terms of the idea of connecting up different types of information, which is how ecologists tend to look at things. But also that I'm one of those people that can eat as much chocolate cake as I want. Okay. Thank you.
Clearly, we're going to have remarks about chocolate cake and ice cream for the rest of the conference, no doubt about it. So let's get started with this preliminary evidence. And our speakers gave us a couple of examples of the kinds of environmental exposures and the interactions with microbiomes that we might be thinking about. Yvonne or John, do you want to just make some opening remarks about this particular topic?
Actually, I really have enjoyed the presentations because I think it has sent several messages to me as a risk assessor. And one of them is very much that diversity is good. Like, the diversity that we see represented in the microbiome is a good thing, and that antibiotics can change that in terms of reducing diversity. While it yields certainly clear health benefits for individuals that need antibiotics, it also reduces diversity that can change ultimately the responsiveness to other things. I think some of the presentations, Stephanie's presentation regarding ozone resonates as well with me, is that trying to understand how we can look at populations and the differences among populations that is based upon their exposure to different types of agents in the environment, such as ozone, in combination with pre-existing conditions such as obesity and ozone, is so complex that from a risk assessor, it kind of makes our head just explode. How do we get our heads around this? And where I kind of view it as a risk assessor is to say that we typically view things as looking at hazard potential, and then we look at exposure dose response potential. And at this point, what I'm seeing is an evolution in our thinking in terms of how we might consider hazard, and that is different populations, different communities may have actually different degrees of hazard because they are susceptible to a greater or lesser extent due to their microbiome or their exposure to things like antibiotics that have changed that microbiome. When we move to dose response or exposure response assessment, that typically takes certain types of data that I'm not sure that I've got my head around how we could approach using these data at this point. That doesn't mean they're not highly valued. Hazard in and of itself is very, very important to us. And so I think as we're looking down the road, I would encourage, in terms of research thinking, that we also consider both characterization of hazard and exposure response, because that sets the stage for us to use information ever more as we look further down. And I think it also helps connect across different types of species. A last comment I might make is that this speaks also to our concept of dose, in that I think one of the readings that was provided before the meeting kind of put the microbiome between environmental agents in the environment around us and absorption. And when you have significant differences in the microbiome, you have significant differences in effectively the absorbed dose. We have not been able to consider that at all. And so this really speaks to the idea of trying to understand the various factors that affect what is dose, what do we mean by dose, and how can we kind of begin to characterize different subgroups or populations or life stages that may be differentially receiving different doses even if the environment is the same. And that may help us understand this interindividual variability as well.
Yeah, so as an academic, I don't dare contradict a card-carrying risk assessor, but I beg to differ a little bit with John in terms of trying to conceptualize hazard as being different by population or microbiome or something. I think hazard is an intrinsic property of a chemical or whatever exposure is, and then the outcome may be different. So I don't think we need to go all the way to the beginning of regulatory decision making and start rethinking hazard. I think that's somewhat been, I think, settled more or less with animal studies, and I would probably leave it there. But what's important, and what I've gathered from this particular set of presentations in our meeting 5 years ago, was that I still feel that we are a community of gatherers, and we haven't started hunting yet, because I think it's fantastic that we have now major efforts to characterize the microbiomes, their changes, their variability, both in animals and in human studies. But I think the next big step has to be taken in studies similar to the ones that we've heard from our colleague in Israel, where there's really parallel collection of all the information that can be collected, and it's longitudinal, and it's in a large enough sample of people. And the issue that we will have to grapple with as the community is to recognize whether these distributions of variability across individuals, if we slice them by genetics, or if we slice them by nutrition, or if we slice them by life stage or some other variable that we know exists there, are they really different, or are they overlapping, partially or largely overlapping? And I think the question then will come up, do we really need to collect all that information, or maybe we can collect or get by with only some of the information for most of the decisions that we need to make? It's not to preclude that science shouldn't be done, but I really am concerned that we make that switch from just characterizing what's out there, and there's an infinite number of possibilities, square inches of human body and billions of people, and gazillions of conditions after application of different health, personal care products and other things, and really start thinking about using whatever scarce resources we have to design proper studies where everything is collected, and then we can make proper decisions in a head-to-head comparison. So in that regard, I think, to me, what most resonated was the presentation that we heard first in this particular session, and I truly believe that that's where we need to go, rather than try to only feel the elephant from one side.
Sharon or Neha, do you want to comment on this particular topic, this idea of how we might use some of this to identify susceptible individuals or populations?
I would like to say something different, but on the same lines. So we've become very good at gathering information. Our tools are now excellent with advancements in mass spec, NMR, and sequencing. We can gather a lot of data. We've begun to mine this data in creative ways. But what we haven't done at a larger or a high throughput scale is to test these hypotheses in some systems. Dr. Matthew mentioned today a quote that all in vitro systems are flawed, but some of them are helpful. And I think some of the new upcoming technologies such as heart on a chip, lung on a chip, lab on a chip, or 3D printing of organs or habitats allows us to test these hypotheses. And that's one area where we should also focus so that we can, all the information that we are gathering and mining can be put into perspective.
So one of the things that was mentioned this morning was that there's an opportunity for us here to begin to look at sort of the ranges of function that are represented by this microbiome that we're able to measure in a number of different ways, and that by looking at biomarkers, we could perhaps get at our arms around what the microbiome looks like, what the range of response of the microbiome might be, and perhaps focusing on critical functions, begin to think about how that might affect certain types of air pollutants or other sorts of chemicals that we might find ourselves involved in. We've always looked for metabolic outliers or other groups from a toxicologic standpoint that would express greater response to the same dose, to get at John's point. And so the question is, can we do similar types of things given what we have now in terms of tools for the microbiome, in terms of sort of beginning to put people into baskets, whether they're those obese people that have the right kinds of characteristics. Is it sex? Is it response to? Is it airway response, for instance, to ozone and things like that? Are there some of those kinds of approaches that might help us? And what are the types of data that we would want to be collecting as we begin to do our studies in order to address those issues? How do we begin to think more innovatively about how we design our studies to be able to collect this information along with the response or toxicology data that we might look for?
Let me get going by just bringing this back to what the status quo or the current practice is. So most of the information that goes into decision making is already collected on models or experiments that are already very heterogeneous in many ways, including the microbiome. So whether it's epidemiology data, whether it's animal studies, frequently you're looking at studies from different laboratories, different time, slightly different exposure scenarios, even in animal studies. So I think as far as general unification of all of those variables that we could be worrying about and not sleeping at night already have been included. Where I think the challenge lies is in trying to make decisions about compounds or exposures or scenarios where we don't have the benefit of these, controlled or uncontrolled observational or clinical trials. And there, making sure that we include the right measurements and our models include the right parameters that could be variable by individual, by other factors, is going to be the greatest impediment, I think, for us moving forward unless we figure out the right models to actually collect that information. So, John, do you agree with that? I mean, can we start separating, things on which we have a mountain of data, and perhaps say that, microbiome probably was one of those variables that already, determined this response. So maybe we may or may not want to go and dig deeper into that. But for the newer chemicals with less information, maybe that's a bigger opportunity, but also a challenge.
I think that it's one of those things that perhaps we, looking back, wish we had more information for the last 30 years, that we didn't collect this type of data.
But you still made a
We
decision.
still, well, of course, and
Right.
it goes back to what difference does it make? That was one of the questions that came up this morning. Well, so what if your microbiome is different? I think we saw some illustrations that it can matter, but nonetheless, we still see the type of data that we can use without information about the microbiome. I think what it does help us understand is fundamentally, people are different. This was the workshop that was held at the end of September, early October, I guess, was there's differences that we can't understand. We don't understand why people are different or why populations or subgroups or even life stages are different in some cases. And this, I think, gives us insights to help understand that, in fact, for some endpoints, there's probably some populations that are exquisitely sensitive. And we may not be well understanding or characterizing or considering that. And so it is important for us to recognize that there's many different factors. This is where the sort of the ecological thinking comes in. There's many factors that influence nutrition, life stage, different types of microbiological influences. And those factors are not well understood in many cases. And so we find ourselves looking at information that we can work with. We do work with that, but we're still, I think, benefiting from ever more information to better characterize those uncertainties. And at least, again, this goes back to sort of at least we understand that we may not be fully characterizing the hazard to populations or to certain groups. And I'm thinking about hazard, not about chemical hazard, but just hazard in terms of autism or other types of outcomes as well. And that may be, in fact, extremely important when we communicate this to decision makers, saying, we understand certain types of factors that influence health, and there's much that we don't understand. I think this supports additional research, actually, as well, support from NIHS or NIH.
Sharon, your example was one where, Stephanie, excuse me. Stephanie, you had shown us an example where we had an observation of obesity and impact on response to ozone. Yet there was a lead there that got you into looking at the interaction with the microbiome, and a hypothesis now that suggests that the microbiome may actually be playing a role. How might we generalize that kind of a thinking in able to design our experiments or think about how we might collect information for other situations where we might begin to look to the microbiome?
So there's a lot of pieces to that question.
Sure.
So I think I want to start just by making a plug for people thinking about obesity a little bit more. 35% of the people in this country are obese. 75%-ish are overweight. What we know about in mice is that makes a huge difference, And it's not just in how big the effect is, it's also in how it comes about. I don't know if you noticed in the data, but in my lean female mice, you give them an antibiotic, they get more responsive to ozone. You take an obese mouse, you give it an antibiotic, it gets less responsive to ozone. So it isn't just that the obesity is impacting how big the response is, it's totally affecting what is causing that response. Now, there's probably a ton of data about how ozone impacts the lungs. I mean, you've made all kinds of recommendations about how we keep our ozone low in this country based on that kind of data. A lot of it is, maybe all of it, is derived from data that was generated before there was an obesity epidemic in this country. And so we make a lot of decisions about what to do based on lean people and what happens in lean people. And most of the people in this country are not lean. And so I just think that is something that we should always be thinking about more. I don't know that it's thought about enough about how obesity is impacting the response to all kinds of things in this environment. As far as the microbiome goes, I think we have an opportunity, to start collecting all kinds of samples, fecal samples, skin samples, from all the large epidemiological studies that we're doing. I'm not yet sure how we're going to use that data in the future, but if we, like in our study, if we can start to figure out which bacteria it is that are responsible for these effects by using the different obesity, different sex, different kinds of conditions and how antibiotics impacts the response to ozone, and we can actually come up with it's these kinds of bacteria or these kinds of bacteria that do it. And you can devise then, simple analyses, on which ones are present, which ones are not present, that would allow you to determine response. I think we could mine a lot of the data that we already have a lot better if we had that information.
So,
Sure,
so
thanks,
Stephanie,
Stephanie.
I agree with you 110%. What I wonder about what the next step would be. Once you know, imagine you know which bacteria are important. What is the next step? Is it an intervention? If you have a drug, in the example that Dr. Rodinbo has, demonstrated this morning, this is a drug. There is a, benefit consequence to using that drug, and you're trying to alleviate some effects. If we're talking about environmental exposures, we're not really trying to, prevent people from being exposed rather than treat them with a drug that can make them better and still go to work. I think, thank God, in this country, occupational health and hygiene has moved on. So once we have the information on the full characterization, have you started thinking of how that will be used in the public health sense? Because when I'm searching for answers, I'm having a hard time understanding where would you actually apply and what would you change. So knowing more is always good, but is that actionable intelligence or else?
Yeah. So, I mean, from a public health standpoint, from a sort of environmental exposure standpoint, I don't know the answer to that. But what has motivated me from the beginning is that obese people get asthma, and their asthma is not treatable by anything that we have in our arsenal right now. If there are things about the microbiome that lead us to the point where we could treat them with a certain class of probiotics or a certain dietary regimen of prebiotics, or even, theoretically, with fecal transplants, that would lead to an amelioration of their disease, that in and of itself, to me, would be great.
So, Stephanie, it seems like there is a parallel there with the idea that, you knew that there were changes in the microbiome associated with obesity, and so that gave a place to look.
Yeah.
Okay. We know that for certain drugs, we're beginning to understand that individuals or populations who take certain drugs are going to have certain changes. We know in terms of populations, but now we're beginning to learn a little bit more about functional changes in their microbiomes and beginning to overlay how some of these xenobiotics are going to be handled. We may begin to look at some of those, and either through changes in lifestyle or other sorts of things like that, address this question of susceptibility to an environmental chemical. And so just as your hypothesis sort of shows the connections and suggests that there may be some interesting opportunities here for us to look at how we can address this particular population, I think there are parallels with other xenobiotics. Questions or comments at this point, John?
I'd just like to make one comment, and that is the comment you made about the controlled human exposure studies for ozone. And Linda Burma might, or Ron might also. I don't believe that we've used obese subjects. Most of the subjects in those studies at EPA have been done in healthy young college students for the most part. We have some susceptible populations, but do you recall if we had any with a higher BMI? Was not an exclusion factor. Okay. Linda was saying that BMI was not an exclusion factor necessarily, but yeah, but it would be very interesting to go back and look at some of these data. Yeah.
And there's a discussion that started on
right. That's right.
People are probably fatter now.
They are. Yeah, Joel Schwartz just said that there's current ongoing studies, and perhaps the population being studied now is larger to BMI compared to where they were back in the 80s.
So I kind of just want to comment a little bit, which is I think it's going to be very important for us to look at the forest instead of all the trees at times. Looking at the trees helps you understand one thing, but looking at the forest is the integrated kind of sum of the whole thing. And I'm wondering, I know when we look at I wonder if all the differential susceptibilities that occur whether it's age, whether it's microbial, whether it's dietary, whether it's lifestyle. If all those things really, we should be, when we look at studies that appear to give us different results, we talk about lack of reproducibility, and it may be because we're not studying the same thing, that there are differences. So when I look at a lot of the epidemiology studies, and there's this whole push to do meta-analysis and pool everything, I think what we often lose is the power of the analyses. I mean, I'm assuming that the individual studies could be strong enough, because we then, we don't acknowledge the heterogeneity in the studies, which may be the reason that when you try to pool them, you kind of lose your effects. And I think we have the same thing with a lot of the animal toxicology studies. Stephanie, you were very clear in saying that one of the reasons you're having very clear data is that your animals are all in the same room, from the same moms, all this kind of thing. And I think that is not done routinely. And I think often the lack of quote reproducibility is more a function of the lack of standardization across a wide range of parameters, most of which we're just now beginning to even consider.
We have just a few minutes for questions. We have a couple of people that are up at the microphones, and let's go ahead and take one over here.
John Arias, NIH again. Clearly, this field has to move from sort of a correlative state to causality. That's obviously where you want to go. And it also strikes me that the complexity of the systems is going to make that very difficult because of the variety of microbes involved and conditions. And so given that, it's sort of reminiscent of where the field of transcription was a number of years ago with the bioinformatic revolution. A lot of data collected looking at relationships in developmental pathways, for example, in response to xenobiotics, trying to make sense of it. But ultimately, you're going to have to look at it from the standpoint of a dynamic system change. So not static points that are so separated that you're going to lose the dynamic qualities that may be revealing. And secondly, you're going to need a reconstitutable system, a testable system. And I think that means you're going to have to start looking at, just genus comparisons and start to drill down to molecular aspects of the metabolomes of those bacteria involved. And that's going to require, a model system that's going to be amenable to such changes. I think the human data are going to be very informative. Maybe interspecies comparisons will help winnow things down, but ultimately you're still going to have to get down to an animal model system that will allow you to sort of, separate the trees from the forest in this case. I just want to make the point.
Thanks. Please.
Yeah, Ann Summers, Georgia. I'd like to get your input, the audience's input too, on the role of ex vivo systems, because certainly there hasn't been any problem with kind of taking lymphocytes out of cancer patients or the general population and culturing them in vitro and asking questions about all kinds of things from drug susceptibility to toxicology. And that's obviously still very complex, but the Shaler flora was a model a long time ago that has primarily been used in vivo with colonization of gnotobiotic mice, but people are beginning to do Shaler flora experiments in vitro, basically. And I'm not saying this is a good thing or a bad thing. I'm just wondering, as a microbiologist for millennia, microbiology, the gold standard was the pure culture. Mm-hmm. We can really find out what's going on if we have the pure culture. And we were all thinking, that's nonsense. These are multiple cultures now. I mean, they always were. They were communities. And microbiology gradually moved towards being able to think about and extract data from small mixtures of cultures. And they may be naturally occurring ones or composed ones. And I just want to put that out there, since the Shaler flora does have a history in this area, and ask, are there things that we should be thinking about that we could learn that would be extrapolatable, in fact, to the very easily extrapolatable mouse model that's already there, by doing things in vitro? biochemical experiments, exposure experiments, different conditions. I mean, it does require what Carl was cheerleading for a little while ago, and that's more people trained in anaerobic microbiology.
Sure.
But I don't want to stop there, actually, because, of course, the skin is the body's largest excretory organ and in a huge area of interaction with the world. We've got material scientists. What's the next generation skin that could be colonized by microorganisms and studied in an ex vivo model for the skin microbiota? So if you all say this is crazy, that's fine. I just thought there might be some useful ideas here.
So, Ann, I'll just start. I think two of our speakers this morning actually talked about the opportunities for developing those, and we're going to have a session tomorrow about future research that might be needed. And I think some of these in vitro or ex vivo systems that you're describing may be very useful directions to be looking at.
It would reduce the complexity in a way that I think might be useful, more than we realize.
Right. So while I think conceptually none of us disagree that this is, one very good way to go, maybe including, human on the chip, whenever that happens. And we had a workshop on this, I think, a couple of months ago. And I think all of those systems are great, but it's very difficult to get them to be just right. I think the Goldilocks problem is always going to be there. And one of the ways I think regulators and researchers now are trying to bridge that divide, in all models being wrong, some being useful, is trying to really understand what questions can be asked with those model systems and what questions you better not even try to pose to that model system. And my personal opinion, again, following this particular field as an outsider, and I may be completely off kilter here, is that we haven't figured out what questions we can answer with microbiome data to start with. And I think once we know a little better as to what microbiome data is good for, then thinking about what simplified models may actually be applicable and okay, then I think that without understanding what questions we can answer in real in vivo situations, either animals or humans, I don't think we should even, contemplate thinking of what next in vitro model would be right, because it will be very easy to get things wrong completely.
So let me go through the final question that we decided we would talk about here and provide what I think is going to be a very simple answer. We all came into this meeting looking for an opportunity to understand the contribution of the microbiome, and in quantitative terms, in a way that could help us put that in perspective with other sorts of things like host genetics and that sort of thing. And I think it's very clear from what we've seen that we're learning more and more, but we don't know at this point. We really don't. And there certainly isn't enough data for us to model a generalized situation at this point. We may be able to get to specific areas where the metabolome may play a very large role with regard to microbial metabolism, and we can begin through pharmacokinetics or other sorts of things like that, understand the contribution in quantitative terms, but that would be the exception. So I'm going to see if anybody wants to disagree with me on that last question, but I think it's pretty tough for us to be able to talk in quantitative terms about the contribution of the microbiome at this point. Yvonne, I'll let you say this, but I'll ask the others to be thinking about a final comment, because we're going to wrap this up in just another minute or two if you have any final comments you want to make.
Right, Bill, I agree with you, but it doesn't mean that we shouldn't be thinking about study designs that would allow us to do that. And again, I think we all would agree that Dr. Elenov's, Uber study, especially with the follow-up component where you prospectively actually test some of your models, that really shows a great case example of what types of studies we should be thinking about, and layering on environmental or drug or chemical exposures rather than just diet and other factors. So I think this is a very exciting path forward to take.
I think the one takeaway, of course, from Dr. Elenov's presentation was that the focus was on individualized or precision toxicity, metabolism, medicine, however you want to look at it, but it was not from the standpoint of populations and dealing with risk issues from that standpoint. Now, in the future, maybe those can be teased out of that kind of a study. So let's just sort of walk down the line here just for any particular comments. John?
I think the comment I would leave is that from a risk assessment point of view, this gives us insights on individual variability and susceptibility. And I think it does lead us to recognize that when we're interpreting studies, there may be significant differences among populations, such as for arsenic in Taiwan versus Bangladesh, Chile, Western United States. And there's differences, and those differences are true differences in terms of either the dose or the population susceptibility that we need to recognize. This may contribute to our understanding of that.
Thank you.
I think what microbiome can do to us can be very wild. The wildest example that I have read about is microbial tryptophan metabolites can cross our gut, reach our brain, and affect our behavior. So it's very important to ask specific questions, but if we are looking at the overall effect of our microbiome, we do land up with surprises like these, where microbiome can really affect how my behavior is or how I respond to certain situations. It's important to ask specific questions, but it's also important to look at more global effects of our microbiome. Thank you.
Stephanie?
So I'm in Washington, so I have to stay on message, okay? Don't forget about all the obese people.
Thanks. Yvonne, last comment? Okay, thanks. Thanks to all of our speakers and discussants, and we'll wrap up this particular session. Thanks.